So looking still at cellular respiration, the overall reaction that occurs in every single cell to get energy in a usable form, but keeping in mind that not all parts of respiration happen in every single organism. So some organisms only do glycolysis. We're moving on to talk about the last stage in eukaryotic organisms, because this will be the electron transport chain is found along the inner membrane of the mitochondria. Before we get to the electron transport chain, we want to think about how did we get here. So we started, quick summary, we started glycolysis with a 6-carbon glucose. We then broke our 6-carbon into 2 3-carbon pyruvates. We got a net of 2 ATP and 2 NADH per glucose. In the citric acid cycle, now we do need to keep in mind that we actually got 2 NADHs before during the oxidation of pyruvate. We also released, really to make this diagram accurate, we released 2 CO2s before. That's all during the oxidation of pyruvate. We're in the mitochondrion now, and we release 4 CO2. We get a net 2 ATP, 2 FADH, and 6 NADHs. So at this point, entering the electron transport chain, we have released all 6 carbons. We have gained 4 ATP. We have 10 NADHs and 2 FADH2s. And we're actually in the matrix of the mitochondria, so the innermost part of the mitochondria. So when we look at the electron transport chain, we want to talk a little bit about what is really going to happen in the electron transport chain. So the electron carriers, NADH and FADH2, are actually going to be oxidized. So they're going to lose their electrons. So our electron carriers transfer the electrons to proteins embedded in the membrane. The inner membrane of the mitochondria. So let's talk a little bit more about this embedded membrane, and you can see it down here in this diagram. These include transmembrane proteins that, redox, so both get reduced and oxidized as they pass electrons. And so when is this happening? This is happening after glycolysis, after the oxidation of pyruvate, and after the Krebs cycle. This is on the inner membrane of the mitochondria. And here's the big question. Why is this happening? Because we have high energy electrons. That are going to be transferred to 3 different proteins, and then to the final electron acceptor, oxygen. But why? Why does this happen? Why do we transfer these electrons? This energy is going to be used to build a proton, not protein, but proton gradient. I'm sorry, I'm kind of writing on the space there. We're going to use this energy to build a proton gradient. Why? Because this gradient will catalyze the formation of ATP. So we're going to look a little bit more at that right now. So, to use the electron's energy, we have to really be aware of what can we use the electron's energy to do. Well, energy does work. So what we do is we use the electron's energy to pump hydrogen ions, which creates what we call an electrochemical gradient. So if we look at this diagram, we see that we can't see that we're using the electron's energy, but we know that the electrons, as they're being passed, that energy is changing the shape of this protein to move a proton. Little word game there. The energy of the electron coming off of the carrier NADH, the energy of this electron changes the shape of the protein, pumping hydrogen ions out. So we are creating a gradient. We have a high concentration of hydrogen ions. What does that actually mean? Well, we know that the concentration of hydrogen ions is actually the pH. So this means that the pH is actually very low. We're actually an acid out here. We have a low concentration of hydrogen ions in the matrix. So this means we actually have a higher pH. This means that the matrix is actually very basic. Move the picture up a little bit. Okay, so we're using the electron's energy. And the structure of the mitochondria is actually what allows us to do that. It's that inner membrane that creates these two compartments. The inner and the outer membrane create these compartments. We're able to create the hydrogen ion gradient because of those membranes. And then, of course, we also have a lot of folds. So keeping in mind that it's not that this is always right here. ATP synthase does not have to be right here. These three need to be next to each other. And they can repeat. So we have created an electrochemical gradient, a high concentration of hydrogen ions. How did we do that? We use the energy of the electron. And so every time it reaches one of these transmembrane proteins, we pump protons, which is another word for a hydrogen ion. Because hydrogen, when it loses its one electron, it's just a proton. So that's why we call it a proton. So these transmembrane proteins, when they receive the electron, use that energy of the electron to change its shape and pump hydrogen ions out. At this very last enzyme, we're actually going to pass the electron to oxygen. Oxygen will get reduced because it receives the electron. And when it receives that electron, it forms water by forming with those hydrogen ions. We do need to know one more thing. Why does the electron transfer from one to the other? It transfers because we actually have increasing electronegativity. Increasing electronegativity. So if I were to ask you which of these was the most electronegative, well, the oxygen. But then this one would be less electronegative, because this Q is going to have to take the electron from here. And then this would have to be more electronegative to take the electron. Site C, more electronegative. This third one, more electronegative, and oxygen the most out of these things. So the electron transport chain itself is all about using the energy to pump hydrogen ions out, create that electrochemical gradient. Once I have the electrochemical gradient, there is actually only one place that is permeable to hydrogen ions in the mitochondria. ATP synthase is going to be a protein transmembrane across the membrane that is permeable to H+ ions. They are going to diffuse through the ATP synthase. And as they diffuse, they actually cause a rotation in this protein, and the rotation results in kinetic energy. Kinetic or physical movement energy, and that physical energy is what creates ATP again. So it's strange, we go from chemical energy to physical energy to chemical energy. So the electrons never get to ADP. The electrons are not getting into ATP. The electrons build the gradient forms kinetic energy, and that's what forms ATP. And this type of ATP is called oxidative phosphorylation. It actually doesn't have very much to do with oxygen, except that it's powered, right? Oxygen's electronegativity is powering all of this. So maybe that's for the oxidative, or maybe the redox reactions. So the process of these moving through is actually called chemiosmosis. A little bit confusing because water is not what is moving through. Hydrogen ions are moving through. Water is formed over here. So just think of a chemical gradient and the movement of the chemical gradient, and that chemiosmosis turns this enzyme physically. Kinetic motion causes the creation of ATP. So taking a second to really look at what's happening here. I have embedded proteins. I'm going to transfer electrons, pump hydrogen ions. The high hydrogen ion, let's go back to this idea of pump. Because normally when we talk about pumping hydrogen ions, we talk about active transport needing ATP. We don't need ATP here. We use the energy from the electron to do the pumping. These pumps don't use ATP. These pumps use electron energy. Electron energy pumps the hydrogen ions. When they flow down their gradient, that's when the kinetic motion will result in the formation of ATP. So, some important points. We've talked about all of these except the first one. FADH, this electron carrier, drops off the electron at the first protein. This means there are hydrogen ions pumped 3 times. NADH actually drops off the electron at the second, which means that hydrogen ions are only pumped twice. So if I look at how much ATP is created, there's actually a ratio of 3 to 2 ATP created for every NADH and FADH2. So we've already really talked about increasing electronegativity, already talked about active transport, electron energy, not ATP providing the active transport, and then passive transport as well. And that is the electron transport chain.